Okay, good afternoon to everyone. In continuation to our previous practical class, today we are going to deal about new practical, that is about preparation of antigen. So preparation of antigen, and generally you might have studied antigen in your theory class. So generally when we are talking about antigen, its definition tells that it is a non-self and that particular molecule must be able to trigger an immune response. So these two attributes tell that non-self means they are not of our own body, but there is an exception that when our own cells they are not behaving like a self, then they also consider as non-self, and a prominent example is cancerous cell. Similar way, when they enter inside the body, they must able to trigger an immune response. Means they should stimulate our immune system to produce some molecules or immune response against that particular molecules. Then only they are known as antigen. So that's why if you will recall the theory class, there is a one prominent statement in generally asked in your exam that all immunogens are antigen, but all antigens are not immunogen. Okay. So all immunogen are antigen, but all antigen are not immunogen. It means that there are two words, immunogen and antigen. Okay. So immunogens generally they are considered as a complete antigen. They are able to show the two characteristic. One that is able to bind with the molecules of immune system, and second uh, uh, second one, that is they must trigger immune response or immune system to produce immune molecules. In contrast to immunogens, if we are talking about antigen generally they are capable to bind with the molecules of immune response, but they themselves are not able to stimulate the immune response. Okay, they require some conjugation or they require some additional procedure before acting as a immunogen. But in general sense, in day-to-day life or in routine book, you can use these two terms very interchangeably. That's why we are commonly use the word antigen. But in immunological sense, immunogen is considered as a complete antigen that is able to bind as well as able to stimulate the immune response. You might have studied in detail about these attributes of different types and features and factors of antigen in your theory class. For example, under type, you might have studied that to whom you can say this super antigen, okay, or to whom you can say heptan, immunogen. Yes, these are the important terminology. Similar way, you might have also studied about the general features of antigen. So general features of antigen that may vary, or to whom we can say the ideal antigen. So if you can see, then size, shape, the structure, complexity, and the chemical nature, whether they are able to degrade or not, foreignness to host. So all these features, they will generally help to decide that how strong that antigen could be in particular host. The last that about the factors determining, which is factors will determine the immunogenicity of that particular antigen. For example, the route by which you are administering the antigen, that is also playing important role because their processing is different, and as the processing is different, the immune response might be different. So similar with dose, host, and the physiological status, the environmental factors. So these all are playing some important role in deciding the immunogenicity of particular antigen. So this is the background about to whom we can call the antigen and how it is different from the immunogen. Now with this background, we will see the next one that, for example, this is the schematic diagram I had kept. So this is the antigen, you can see yellow one, okay, that has four, 1, 2, 3, 4. These are four different epitopes, okay. So the definition like epitope, paratope, okay, agritope, it must be clear in your mind if you don't have any idea about this. So here this antigen that has 4 different epitope means 4 type of antibody are supposed to produce when they are inoculated in some host. So for example, this antigen once entered in mice, then mice will recognize this antigen, and in response to that antigen, as it is a non-self antigen, non-self molecules, that will stimulate protein molecules like antibody against this one. And generally antibody are targeted against this 4 epitope. So that's why if you will see the serum contain antibody 1, 2, 3, 4. That is a mixture of antibody against this one. The second scenario, if you will see, in second scenario, this this will bind with antigen. Okay. Now then in second scenario, what will happen? All these 4 antibody, if we are capable to differentiate clone, and we are capable to differentiate all these 4 antibody in different tubes, then a particular antibody is specific for specific epitope. Such antibody are generally known as monoclonal antibody, whereas the mixture of antibody that is not the specific, they are considered as a polyclonal antibody. Okay. Now, so it is clear to you that to whom we can tell the polyclonal antigen and to whom we can call the monoclonal antibody. Okay. Now antigen-antibody interaction. So antigen-antibody interaction, that is also an important aspect, because all the serological tests, or you can say all the practicals that we are supposed to cover in immunological classes, they all are based on the principle of antigen-antibody interaction. So antigen-antibody interaction, if you can compare, then it is just like lock and key. Okay. So if you want to open or unlock this lock, then even though you have thousands of key, you cannot able to open this lock unless until a specific key is with you. Okay. So there is a specificity between the key and lock. A similar example is also available from biochemistry. If you can say enzyme and substrate. Okay. So enzyme and substrate, they are the specific one. Specific enzyme act on specific substrate along with some requirements of environment and catalyst. And once they met all the condition, then they will lead to the product formation. So this is also specific. In similar way to these two examples, if we can talk about antigen and antibody, so antigen and antibody, their binding, that is also specific. It is not randomly bind to any antibody. Okay, so specific antigen or epitope can bind to specific antigen antibody site only. So in nutshell, antigen and antibody are 2 important molecules of entire serology. Okay, so if you know any one of these 2, either you have the antigen or antibody, then you can easily develop the diagnostic kit and you can easily use them in a therapeutic aspect. So that's why we required particular antigen or antibody, so that corresponding unknown molecules we can easily find out from the mixture of that particular molecules. So this is the basic concept of antigen and antibody interaction. Now coming to today's class, that is about preparation of antigen. So we'll deal into 2 ways. One, that is preparation of antigen from bacteria, and second, preparation of antigen from viruses. So bacteria, I would like to quote this figure. This is well-known figure. You might have seen in many books and many presentations. So this is a classical pictures or diagrammatic representation of bacteria with all sort of their accessory and necessary structures. So all bacteria, they contain cell membrane, and cell membrane antigen, generally they are quoted as somatic antigen, and generally somatic antigen is designated as capital O. Okay. Similar with capsule, there are some bacteria, they contain the capsule, and those bacteria, they may able to produce the capsular antigen, and generally capsular antigen is designated as a K, capital K. Now many bacteria, they are able to produce the toxin. Okay, so once we are able to able to harvest that toxin, and that toxin can be inactivated or can be used as an antigen after inactivation. That is generally known as toxoid. Okay, so this is the features of some bacteria. Now, there are some bacteria, they are considered as a motile, and that motility is principally because of presence of flagella or sometimes fimbriae. So fimbrial antigen, that is designated by capital F, whereas the flagellar antigen, that is flagellin, that is the principal protein component of flagella, that acts as an antigen, that is designated as capital H. And below this one, I have mentioned the method by which we can isolate that particular antigen. In brief, all this technique, whether it is alcohol treatment or formalin treatment, or you can go for heat treatment or acid treatment or any other method, the three things you have to keep in mind. The first, it must be in native form. Okay. Second, it must be pure, as much as purity you can mention, that must be purity. And third, the criteria of cultivation is vary, and that generally depends on at what time particular antigen is producing in maximum amount. Okay. So this is about this is in brief about different types of antigen present in bacteria. Now we will see one by one. Generally, the antigen is prepared from or prepared, that is known as bacteria. Bacteria, in simple word, we can say that they are the killed bacteria, and that killed bacteria can be directly used as a antigen. Now, the procedure for production of bacteria is like this. The first and foremost step, that you must have a pure culture, okay, and that pure culture, if it is stored in your laboratory, then you require to revive that bacterial store from stock culture, and you can identify whether they are pure or not, and they are okay for proceeding further or not. The second step, you can say bulk production. Bulk production and harvesting, because antigen preparation requires the bulk volume of bacteria. So whatever we are performing in laboratory, they might not be sufficient to produce the sufficient amount of antigen. So for that we require the production of bacteria in bulk. And in laboratory we may achieve this by growing bacteria in growth, or at the industry level they may cultivate bacteria using big fermenter. Okay? So they will get the thousands liter of antigen in a single batch. After production and harvesting the specific bacteria, then we have to go for inactivation. And inactivation can be achieved by different methods. First, that is by heating that particular culture at 30 minutes at 100 degrees centigrade. Or you can go for formalin inactivation by adding 0.5% formalin in final volume. And you may go for alcohol treatment. So that depends on your starting material and the use of that antigen in your future. Okay. Now that will be followed by the washing. So once you will go for washing, so whatever non-required materials were there, they will get washed out, and at last you can suspend them, that is particular to 0.4 methylene standard. So that will give a specific concentration of antigen per ml in your final lot. That final lot may need to be add some preservative if you want to require to preserve that antigen for longer duration, or you can go for storage of that particular antigen at low temperature for some time. Okay. So this is general procedure for preparation of bacteria. In similar way, if you want to prepare the hemolysin, okay, then generally it must be before seeing the procedure, it must be clear to your mind that to whom we can say the hemolysin. Hemolysin are generally the antibody directed against CPRBC, and generally this hemolysin generally used in complement fixation test as a detecting platform. So in that case, we require CPRBC as well as the antibody against that SIP RBC, which are capable to lyse this RBC once they get infected. So if you want to prepare the SIP RBC as an antigen, then you have to collect the SIP blood from healthy and disease-free SIP in special solution that is known as elsewhere solution. Elsewhere solution in equal proportion you have to collect that one, and specifically it is recommended to collect blood in elsewhere solution because that contains some chemicals that will make RBC survive or live for longer time. Once you had collected your sample, then you have to go for pelleting of RBC, and generally it is achieved by centrifuging that blood at 2000 rpm for 6 minutes, and preferably at low temperature. So you will get the pellet at the bottom, generally that is known as packed cell volume, and whatever supernatant remain on the top, that is supernatant or plasma, that you have to discard, and you have to give that pellet 2 or 3 washing as per your need with PBS or normal saline, so that you will get the pure RBC without any plasma protein or any other unrequired materials. So at the last washing you will get the packed cell volume, and based on that packed cell volume you can prepare the required percent of RBC. Sometimes it may require 0.5% or 1% or 2%. So you can prepare this one, and then you can go for either storage of this RBC, or you can go directly for inoculation of that RBC as an antigen in animal in which you have to raise the antibody. But on storage, it has limited lifespan, so you cannot store for longer time. You have to use it very immediately, and preferably it is better to collect the fresh sample of fresh RBC for production of hemolysis. Now, the next and little bit complicated, that is a recombinant protein, or you can say biotechnological based tool for preparation of antigen. And this is particular important because we are avoiding unnecessary proteins, because all the proteins they stimulate immune system in different way, but all proteins or the antibody against small protein are not of our importance. In such cases, what we are trying to do, we are by utilizing the biotechnological tools, we are removing unwanted protein, and we are targeting the specific protein that is of our interest, and that will produce the maximum antibody that is provided, or that serves our purpose. So first step in that one, that is bacterial transformation. So bacterial transformation means whatever gene of our interest that encode the protein that is able to secrete the antigen that is of our target, that we need to coat inside plasmid one. Okay. So once you will coat here, then this particular plasmid we need to transform in bacteria. And this transformation, for example, this is a competent bacterial cell, this is a recombinant plasmid. Now once you will go for heating method, then this plasmid will take up by bacteria, and this bacteria now considered as a transformed bacteria, or you can say recombinant bacteria. Now in the next step, we have to differentiate whether bacteria are really transformed or they are the non-transformed bacteria. So what we are doing, this one is a non-transformed and this is a transformed one. So we are going for plating in plates that have some selective media. So here selective media, that is antibiotic. Okay. So if antibiotic, without antibiotic plate we are taking, that all the cells, whether they are recombinant or non-recombinant, all will grow. But if you want to grow, if you want to grow only a transformed bacteria, then you have to add the antibiotic. So in presence of antibiotic, only transformed bacteria can grow. So you can say all these bacteria are transformed bacteria. There is no untransformed bacteria like this. Okay. So once you get selected, now this particular gene is inside the bacteria. Now you have to go for expression of that particular gene. So in expression, what we are doing, we are providing some media in which we are giving some induction signal. So this induction signal will stimulate bacteria to produce the protein that is encoded by our plasmid. Okay? And that plasmid carry the gene of our mutation. So now, once they will produce, then we have to check whether they produced or not. So generally, this production we can check by SDS page. Okay, so we can tell that okay, our target protein is produced inside this transformed bacteria. Now, once this is if you can see, this is performed in laboratory at smaller scale. But we require to produce the antigen in larger scale, then we have to go for scale up. Okay. So in scale up, what we are doing, we are going for big culturing in big flask. That is generally known as fermenters. Okay. So in fermenter, we are growing this bacteria. We are inducing this bacteria to produce that target protein. And once this target protein produced, we can go for purification, or you can say isolation of that protein. And generally isolation is achieved by chromatography. So chromatography that will specifically bind our target protein, and remaining proteins get washed out, and at the end we will get our target protein that can be used as an antigen. And this purity of this target protein can be seen by chromatography further, or you can say HPLC method. So now we will get some purified protein or antigen. Okay, the purified protein or antigen that can be used as an antigen either for your diagnostic platform development, or you can use that recombinant protein for therapeutic aspect. So this is in brief about preparation of antigen using modern technology like biotechnology or molecular tools. Okay. Now we will see the summary of this one. The summary is very straightforward. You can say the first step that you have to go for seed stock revival. Okay, this is the first step of any antigen preparation. Second step, you have to go for pure culture and scale up. Means you have to produce pure culture method. Then third, you have to go for bulk production utilizing a fermentation process. Then after production, you have to go for inactivation. This is a fourth step, that is inactivation. Inactivation can be achieved by heat method, or it can be achieved by some chemical method like addition of alcohol or formalin or some other chemicals. And once you can inactivate, then you can go for packaging, or you can say addition of preservative, and then storage. So these are the 4 or 5 steps that is generally followed similar way for all type of antigen. Now the last, that is about viral antigen preparation. So if you want to prepare a viral antigen preparation, then you must it must be clear in your mind the composition and structure of bacteria and virus. So we had already shared the slide for bacterial structures. Similarly, if you can see the viral structure, okay. So virus generally at the core, okay, you will get the nucleic acid. Okay. Any virus if you will take, at the core that has nucleic acid, generally that is DNA or RNA, and sometimes they are associated with some proteins like this, okay, that are viral coded and they require for their replication. This particular nucleic acid is covered by this type of proteinous covering that is known as capsid. Okay, and this capsid generally they are proteinous in nature, viral encoded, and these proteins are important. These proteins are important for stimulating immune response, and That's why they are considered as a good viral antigen. The second viral antigen, that is, if you can see there, some viruses they have envelope, and such viruses are known as enveloped viruses. So this envelope, okay, this envelope contains some viral encoded proteins. Okay, you can see here. So this viral encoded protein, they are considered as a spike, or you can say peplomer. So this spike protein or peplomer protein, they are directly responsible for binding of virus with a host cell, and generally they are capable to produce virus neutralizing antibody. That's why they are also considered as an important viral antigen. So capsid protein, you can say capsid protein, this is a first antigen of viral, and second antigen, that is a spike protein. So these are the two important protein that can be produced in different way in laboratory as a viral antigen. So in general, the first method, that is a whole viral antigen. The simple way is we are cultivating viruses, we are inactivating, and we are utilizing that whole viral antigen as your antigen in your study. So the steps for production of whole viral antigen. The first, that is a revival of your virus stock, that is the first step. Okay. The second step, you have to go for bulk production and harvesting. Bulk production, again I had mentioned, it is important because we require to produce antigen in large bulk, in thousands of liters. So that's why we have to go for bulk production. The third, that is purification of this antigen. Because generally in bulk production we are utilizing the cell culture or the embryonated egg. So that cell, they are generally of mammals origin, and that proteins need to be separated from viral protein. Otherwise they will interfere in subsequent step. So that's why required purification. And sometimes this may require concentration, because in performing this thing, the concentration of antigen might not be appropriate. So you require to concentrate that antigen either by ultracentrifugation or filtration techniques. So you will get the sufficient antigen per ml of your final lot. After getting this one, the fifth and most important step, that is inactivation of this antigen. And inactivation generally achieved by addition of formalin or the molecules like BEI, or some heat inactivation can also be performed. But the ultimate purpose, ultimate motto is there should be not a single viral particle left alive. Okay, and after this, if required to add some preservative, we need to add the preservative, and then we can go for storage of that antigen. So this is a one way of production of viral antigen using whole virus as a starting material. There we cannot differentiate whether it is a capsid protein or it is a spike protein. Okay, and such antigen generally produce a polyclonal antibody. In contrast to this, the second method, that is known as recombinant viral antigen. This is entirely similar to what we had studied previously, okay, in bacteriological one. Similar way, here just we have to add the gene of viral antigen. Okay, the remaining procedure will remain the same, and at the end we will get some antigen that is considered as a viral antigen. So this is a specific antigen, single antigen, and that can be used for increasing the specificity of some test. Okay. So this is in brief about antigen preparation from bacteria and virus. So at the end, or in the end of this class, I would like to tell you that whatever procedure we are following for antigen preparation, the first and foremost thing that you have to remain in your mind, that is time of harvest. First thing, because antigen may not be constantly expressing at constant rate. For example, if flagellar antigen, then flagellar antigen may be secreted at 25 degree, may be produced maximum at 25 degree in some species, and at the log phase. Then your time time of harvest must be appropriate. The second, that is, your process must be in such a way that will retain the native state. Native state, because if the epitope get disturbed, then you will not get the actual response from that antigen. So your procedure must be such that it must not let any bacterial virus infectious at the end of your process, and it must try to preserve the protein in their native form as much as possible. And the third and the last point, the purity of your antigen. And this purity, to maintain the purity, you have to cautious during all the step of the entire process, right from the isolation of your revival stock, from at the end of your storage and inactivation. So with this, we will end today's class, and thanks everyone for listening me. Okay. So now class is